Hi everyone, welcome to my channel Pure Biology and I am Taposhi Karmakar. Today I come with another video related to immune system, that is RIA or Radioimmuno Assay. Okay, so before going to this topic, I want to say thank you to all of you who have subscribed my channel and support me a lot. So let's continue. Ria and Eliza. What are actually these? Ria and Eliza. These both techniques that rely on the formation of antibody and antigen complex, and both can be used to measure the quantity of unknown protein present within a given sample. Both play a critical role, that is role in diagnosing various diseases for any different types of organisms. So what is saying that Ria and Eliza, these are the two techniques, and here actually techniques means this is a process where we will mix the radio, generally in case of RIA, the radio labeled antigen and unlabeled antigen both are mixed with a solution and then mixed with the antibody, and here by binding of radio labeled or unlabeled antigen with the antibody, we can easily find out the amount of labeled antibody amount of labeled antigen and unlabeled antigen. Okay, so here by this process, so what is the actually role or why these processes are needed for us? Because by which we can easily identify any unknown particles, okay, that create any disease for a host or for a body. Clear? So these two processes generally used for identifying the unlabeled antigen or unknown antigen that creates any disease in the body. Or suppose any human being, he or she is infected any unknown disease, and in that situation we have to identify the particular foreign particles by which they are infected. So in that situation we use serum, body serum or blood serum for the identification of the foreign particles by which these people are infected. Clear? So here the principle is what? Generally it involves the competitive binding of radio-labeled antigen. First of all, some known antigens are used for a particular antibody, and here the antigens must be labeled or radio-labeled to a high affinity antibody, and the labeled antigen is mixed with antibody at a concentration. Suppose first of all the radio labeled antigen and or the foreign particles, but here the foreign particle is obviously known. This is mixed with the antibody. Okay, and then how much concentration is needed? That the saturation of antibody or the antigen in a particular concentration, they are mixed, that means known antigen and the antibody, they are mixed in such a concentration that here all the antigens, they totally saturates by the antibody. Okay, or you can say that the remaining all the radio labeled antigens, they totally bind with the all present antibody. Clear? And here increasing amounts of the test sample, then obviously within that mixture increasing amount of test samples are added that containing unlabeled or unknown antigens. And now what happened? So the concentration of the unknown sample is so much, and does not distinguish the labeled and unlabeled antigen. So, two kinds of antigen compete with each other, and obviously with the increasing concentration of the unlabeled antigen more than the labeled antigen, they displaced the labeled antigen from the binding sites. What is saying there? That first of all the labeled and antibody both are mixing in a saturation condition. After that, the unknown particles containing sample is mixed with that, and here the antibody cannot, obviously increasing numbers of foreign particle must be there, and here the antibody cannot distinguish both of them, that means the labeled one and the unlabeled one. In that situation, the labeled antigen that already bind with the antibody, when the unlabeled antigens containing mixture is added, the amount of unlabeled antigen is so high that the labeled antigen totally displaced from the binding site of the antibody. By what? By the unlabeled antigen. Again I repeat. First, the labeled antigen that binds with the antibody, and when the increasing number of unlabeled antigen mixed with them, then what happens? The labeled antigens are totally displaced by the unlabeled antigen from the binding site of antibody. Okay? So, here what will happen? Suppose this is the actual mixture. Antibody, this is unlabeled antigen, and this is labeled antigen. Okay. So here the labeled antigen that is bind with one antibody, and unlabeled antigen that binds with another antibody. Okay. So here the labeled antigen antibody complex and unlabeled antigen antibody complex both are formed. And obviously, the number of unlabeled antigen antibody complex is maximum. Okay, number of unlabeled antigen antibody complex maximum than this. Here, very short rough structure is just drawn here, and you can say that both are same. No, that's not. Just want to say you that two type of antigen complex are formed, but only we just count both of them, and by which we can just count the unlabeled antigen. So here the only two is seen here generally, but number of unlabeled antigen antibody complex generally maximum than this one, because the number of unlabeled antigen that is present in the mixture is greater than the labeled antigen. So here the number of antigen that is unlabeled with the antibody is greater. Okay. So this is the actual principle of the RIA. Okay, just only principle. Next, another thing related of this RIA also we have to learn. Okay. Okay. So here the radio labeling and collection is actually the two processes are described here. Generally, what kind of isotopes so we use? Isotope, not element. Generally, we can use for the radiation or for radio labeling of antigen. Radio labeling of antigen. Generally, I1 to 5 is used, that is gamma emitting isotope, and another is the 3H, that is the tritium, also used as so generally two type of isotopes we can use. One is gamma emitting isotope, another is beta emitting isotope. So example is I1 to 5, and another example is 3H, that is the tritium. Okay, these are used for labeling the antigens, known antigens obviously. and the decrease in amount of labeled antigen bound to the specific antibody in presence of test sample is measured. So what is generally measured here? The decrease, how much amount of labeled antigen that bound to the antibody in the presence of test sample is measured. So decrease means how much amount of labeled antigens are displaced by the unlabeled antigen, that is actually we have to determine. Okay, and that's why we generally use, in the first step we generally use 50 to 70% of fixed quantity of radio labeled or radioactive antigen. Okay, 50 to 70% of radioactive antigen or radio labeled antigen. Obviously the number of antibody must be less here, number of antibody must be less here or lower than 50 to 70%, you can say. That means if you assume that 100% is the solution, so 70% must be antigen, labeled antigen, and 30% is antibody. Okay, so what is happening here? If the antibody, number of antibody is very less, that's why the unlabeled antigen, they compete with the labeled antigen for binding with the antibody binding site. Okay, so displacement of radio-labeled antigen is possible by the unlabeled antigen as the number of labeled antigen is maximum than the antibody. Okay, so the amount of antibody is very less, that's why the competition between the labeled antigen and the unlabeled antigen is occurring, and that's why the displacement of the radio labeled antigen is possible. Clear? So next step, that is one thing, this ensures, I am saying that this ensures that unlabeled antigen to the mixture will compete with the labeled antigen. Next is the collection, and obviously here the radio labeled antigen is a part of the mixture, and the unlabeled antigen, it is the body serum or blood serum and another body fluids. Okay. Next, the collection. How we have to collect or determine the number of unlabeled antigen. So here first we have to collect the antigen antibody complex from the free antigens. Okay, from the free antigens. Antigen antibody complex are collected first of all or separated from the free antigens, and free, obviously there may be radio labeled antigens also present or maybe unlabeled antigen also present. So in the free cases both type of antigens must be there. Okay, next the the radioactivity in the precipitate is measured. Okay, so separation is possible by the precipitating the mixture, and the precipitate is, the radioactivity of the precipitate is measured. Okay, and this is measured by a standard curve or by drawing a standard curve, and from the plot of the amount of antigen in the test mixture is determined. Or here you can say that the standard curve generally, this is generated using the unlabeled antigen samples of known concentration. Okay, so here the standard curve must be drawn with respect to the labeled antigen. Generally we plot the unlabeled antigen by making the or by doing the curve. Okay, or by drawing the curve. So how the curve is drawn? Generally the curve is drawn by plotting the unlabeled antigen. okay, amount of unlabeled antigen with respect to the labeled antigen. Clear? So when we plot the number of labeled antigen and unlabeled antigen, we obviously have got a standard curve, and by seeing the plotting we can easily determine the number of or the amount of unlabeled antigen. So this is the process of collection, separation and about the radioactivity or the radio labeling. And next step we will learn about another thing that is related to the radioimmunoassay. There is another thing I have to explain. I think that it must be obviously explained when we generally discussing about RIA. Okay, so the thing is that the precipitation of AgAb is possible either adding anti-isotype antiserum, okay, or sometimes use of protein with high affinity of immunoglobulin. It is also used for the separation. In third case that agarose beads generally used. Agarose beads. What is the using or what is the its role of in separation? That is the agarose beads which bind with the labeled antigen. Okay, agarose beads which bind with the labeled antigen. Okay, and in that situation after that we centrifuged and washed and then collect or then we can count. Okay, number 4 is another approach, that is antibody is immobilized, where in the wall of microtiter walls, okay, in the wall of microtiter, not walls, wells, okay, and amount of bound antigen is determined. So another, all processes are obviously in book, how the separation of labeled antigen, unlabeled antigen is possible. So you can check out or follow the books obviously. So but here I just describe the few process how the precipitation is possible or how the collection or separation is possible. Okay, so this is actual radioimmunoassay is explained here.